Now we will be going to discuss what is a gene. Okay, gene. Archibald Garrod and William Bateson concluded in 1902 that certain diseases among their patients, okay, who were scientists A.G. and William Bateson, okay, William Bateson, okay, Archibald Garrod, Archibald, Archibald Garrod. Okay, Archibald Garrod and William Bateson concluded in 1902 that certain diseases among their patients were more prevalent in particular families. Okay, by examining several generations of these families, okay, Garrod found that some of the diseases behaved as if they were the product of the simple recessive allele. Okay, recessive alleles. Okay, he concluded that these disorders were Mendelian traits and that they had resulted from changes in the hereditary information in an ancestor of the infected families. Okay, Garrod investigated several of these disorders in detail. Okay, al alkaptonuria. Okay, alkaptonuria, the patients produced urine that contained homogentic acid. Okay, in alkaptonuria, the patients produced urine that contained homogentic acid. Okay, in alkaptonuria, the patients produce urine that contained homogentic acid. Okay, the patients produce urine that contained in alkaptonuria. The patients produce urine. Okay, urine. Patients produce urine. Okay, that contain homogentic acid. This substance oxidized rapidly when exposed to air, burning the urine black. Okay, turning the urine black. Okay, this substance oxidized rapidly. Okay, when exposed to air, turning the urine black. Okay, in normal individuals, homogentic acid is broken down into simpler substances with considerable insight. Garrod concluded that patients suffered from alkaptonuria lacked the enzyme necessary to catalyze this breakdown. Okay, he speculated that many other inherited diseases might also reflect enzyme deficiencies. Okay, he speculated. Okay, from Garrod's finding, okay, it could be inferred that the information encoded within the DNA of chromosome acts to specify particular enzymes. Okay, this point was not actually established, however, until 1941 when a series of experiments by Stanford University geneticists, okay, George Beadle and Edward Tatum, okay, this point was not actually established, however, until 1941 when a series of experiments by Stanford University geneticists, okay, George Beadle and Edward, okay, provided definitive George GB, this is you can say GB and etc, okay, provided definitive evidence on this point. Okay, Beadle and Edward deliberately set out to create Mendelian mutations in chromosomes and then study the effect of these mutations on the organisms. Okay, deliberately set out to create Mendelian mutations in chromosomes. Okay, Beadle and Tatum exposed neurospora spores to X-rays, expecting that DNA in some of these spores will experience damage in the regions. Okay, Beadle and Tatum exposed neurospora spores to X-ray, expecting that DNA in some of these spores will experience damage in the regions. Okay, DNA in some of these spores will experience damage in the regions. Okay, DNA in some of these spores, okay, will experience damage in the regions, okay. In some of these spores, okay, expecting that DNA in some of these spores will experience damage in the regions, okay, encoding the ability to make compounds needed for normal growth, okay. He concluded that mm, he expecting that DNA in some of these spores will experience damage in the regions encoding the ability to make compounds needed for normal growth, okay. Will experience damage in the regions, ओके। अब तक हमने पढ़ लिया है कि हमने अब हम पढ़ हमने ये देखा कि हमने न्यूरोस्पोरों को एक्सपोज किया एक्सरेज से और इनमें मुख्तलिफ म्यूटेशंस आ गए, ओके। जो मतलब इनमें नॉर्मल ग्रोथ नहीं हो सकती और इनमें मसला आ गया, इनमें अब म्यूटेशंस आ चुकी हैं, ओके। ये देखने के लिए एक्सपोज एक्सपेक्टिंग दैट DNA इन सम ऑफ दीज स्पोर्स, ओके, will experience damage in the regions including the ability to make compound needed for normal growth, ओके। ओके, अब इसको हम DNA changes of this kind are called mutations and the changes that have undergone such and the change and the organism that are called mutants, okay। म्यूटेंट्स उस ऑर्गेनिज्म को बोलते हैं, ओके। DNA चेंजेस ऑफ दिस काइंड आर कॉल्ड म्यूटेशंस, ओके। DNA में म्यूटेशन आ गई और उस ऑर्गेनिज्म को हम म्यूटेंट बोल रहे हैं, ओके। अब ग्रोथ ऑफ स्पोर्स ऑन डिफाइंड मीडियम, ओके। अब हमने देखा कि इनिशियली दे अलाउड द प्रोजेनी ऑफ द on a defined medium containing all of the necessary nutrients for growth so that any growth referring mutants resulting from the irradiation would be kept alive, okay। मतलब इनको पहले सही जगह पे किया गया और इन्होंने अपनी ग्रोथ शुरू कर दी, ओके। लेकिन जब इनको किसी मिनिमल मीडियम पे जो कि शुगर का अमोनिया साल्ट्स और फ्यू विटामिन और वाटर पे मुश्तमिल था, उस पे जब इनको प्लेस किया गया या इनकी उधर ग्रोथ करवाने का कुछ सोचा गया और ये देखा गया कि वो कौन सी म्यूटेशंस हैं और म्यूटेशंस वो कौन सी मेटाबॉलिक डिफिशिएंसीज क्रिएट कर रही हैं। तो इन्होंने ये देखा कि ये जो है वो मिनिमल कल्चर पे ग्रोथ नहीं हो सकी इनकी, ओके। cells that had lost the ability to make compound necessary for growth would not survive on such a medium, okay। Using this approach succeeded Bedell and Tatum succeeded in identifying and isolating many growth deficient mutants, okay। ओके, इसके बाद फिर रिसर्चर ने ऐसे किया कि मिनिमल मीडियम को जो था मिनिमल मीडियम था उसमें वेरियस केमिकल्स ऐड किए। जैसे ही ऐड किया जो कि ग्रोथ के लिए अच्छे थे, साजगार थे, तो इधर ऐसे होने हुआ कि ग्रोथ के लिए वेरियस केमिकल्स, ओके। जैसे कि हमने अर्जिनाइन ऐड किया, मुख्तलिफ केमिकल्स ऐड किए और ये देखा कि कौन सा केमिकल उस म्यूटेशन को सही कर सकता है और नॉर्मल ग्रोथ हो सकती है। जब हमने अर्जिनाइन किया तो उसने ये इजाज़त दे दी परमिटेड सिविल म्यूटेंट स्ट्रेन्स, ओके, डब्ड अर्जिनाइन म्यूटेंट्स टू ग्रो, ओके। बहुत सारे म्यूटेंट स्ट्रेन्स जैसे कि ख़राब जो अर्जिनाइन म्यूटेशंस थी उसमें वो उन्होंने ग्रो करना शुरू कर दिया, ओके। और जब इनकी क्रोमोसोमल पोजीशंस लोकेट की गई तो ये देखा गया कि जो अर्जिनाइन म्यूटेशंस वर फाउंड टू क्लस्टर इन 3 एरियाज, ओके। ये एक इन्फॉर्मेशन है, ओके। तो ये था अब सारा तरीक़ेकार, ओके। तो इसके बाद एक तस्वीर भी है, ओके। What the picture says? Picture says that wild type neurospora minimal medium, okay, wild types, okay, asexual spores, X-ray of ultraviolet light, X-rays of ultraviolet rays, asexual spores, growth on complete medium, okay, meiosis हुई, meiosis पर select one of the spores, okay, product of one meiosis, okay, test on minimal medium to confirm presence of mutations, okay, test on minimal medium to confirm presence of mutations, okay, growth on complete medium, this is growth on complete medium, okay, and growth on complete medium, okay, this is also complete medium, okay, wild type neurospora, and this is minimal medium, okay, minimal medium was transferred to asexual spores, okay, and then these asexual spores were growth on complete medium and sexual and meiosis was done and product of one meiosis, select one of the spores, okay, growth on complete medium, okay, minimal media supplemented with, okay, pyridoxine, choline, nucleic acid, arginine, riboflavin, Okay, minimal control. Okay, thiamine, niacin, and these all. Okay, consider these names, their names. Okay, P and P amino benzoic acid, pyridoxine, P amino benzoic acid. Okay, choline, inositol, PACI. Okay, nucleic acid, folic acid, arginine, niacin, riboflavin, folic acid, niacin, riboflavin, TM. Okay, P pyridoxine, amino benzoic acid, P amino benzoic acid, choline, insotoline, inositol. Okay, P pyridoxine. Okay, P amino benzoic acid, C choline, inositol. Okay, inositol. Okay, pyridoxine. Okay, A amino benzoic acid, P amino benzoic acid, choline, inositol, nucleic acid, folic acid, and folic acid, and arginine. There was also present the arginine. Okay, northern western frontier province. Okay, you can think of that. Okay, Pakistan and China international airport. Okay, Pakistan and China international airport. Okay, okay, northern western frontier province. Okay, the mutations. Okay, the tamam mutations. Okay, okay, now okay, first of all, there was present wild type neurospora. Then asexual spores were done. That this was the minimal medium. Okay, the minimal medium was done here. Okay, and then growth on complete medium. One asexual meiosis, product of one meiosis, select one of the spores, growth on complete medium, test on minimal medium to confirm presence of mutations. Okay, minimal media supplemented with. Okay, this is one of the spores on complete medium, and okay, and this is one of the spores minimal media supplemented with pyridoxine, choline, and inositol, nucleic acid. These all are the pyridoxine, amino benzoic acid, P amino benzoic acid, and choline, inositol, and niacin, nucleic acid, folic acid. Okay, arginine, P. This there the P stands for, I think this may be R. Okay, R. Thiamine, minimal control. Okay. Beadle and Tatum's procedure for isolating nutritional mutants in Neurospora. For isolating nutritional mutants in Neurospora. Okay.